Hello everyone, my name is Jovy Caorte, a Bachelor of Science in Marine Biology student of Visayas State University by campus, and I will be your lecturer for this topic, which is all about biology. For our first topic, we will refresh your knowledge on the field of microscopy. So, microscopy is the science of investigating small objects and structures using a microscope. And microscope, it is an instrument used to magnify objects that are too small to be seen by the naked eye. So list all microscope parts that you still remember. Time's up. These are all the microscope parts. So how many parts did you still remember? Is it almost all or not so much? Don't worry, we will talk about it, about the microscope parts and its function. So for our first part, the eyepiece. So the eyepiece is the lens where the viewer looks to see the specimen. The eyepiece usually contains a 10 times or 15 times power lens. So, the diopter adjustment. So, this is useful as a means to change focus on one eyepiece so as to correct for any difference in vision between your two eyes. Next is the nosepiece. So, it is a rotating turret that houses the objective lenses. The viewer spins the nosepiece to select different objective lenses. Next is the objective lens. So, one of the most important parts of a compound microscope, as they are the lenses closest to the specimen. A standard microscope has 3, 4, or 5 objective lenses that range in power from 4 times to 100 times. When focusing the microscope, be careful that the objective lens doesn't touch the slide, as it could break the slide and destroy the specimen. Next is the stage clip. So, this clip holds the slide in place. The aperture. The aperture is the hole in the middle of the stage that allows light from the illuminator to reach the specimen. The diaphragm. The diaphragm, it adjusts the amount of light that reaches the specimen. The condenser. So, it gathers and focuses light from the illuminator into the specimen being viewed. The illuminator, it is the light source for a microscope. Older microscopes use mirrors to reflect light from an external source up through the bottom of the stage. However, most microscopes now use a low voltage bulb. The head. This is the tube that connects the eyepiece to the objective lenses. The arm is the part that connects the body tube to the base of the microscope. The platform where the slide is placed is called the stage. So, the coarse adjustment is the knob that brings the specimen into general focus. So list all microscope parts that you still remember. Located. So the light adjustment adjust the brightness of the light. And lastly, the on and off switch. And since we are done with the parts and function, let's move to the next stage about how to properly use a microscope. So, turn on the illuminator. When using the dimmer, it is best slowly increase the light intensity as the lamp heats up quite quickly. Then, place a slide or specimen on the stage with a simple directly above the aperture, and if possible, fasten it to the stage with the stage clips. Reminder, a cover slip is always needed to allow for the best quality image. Ensure that the diaphragm is completely open, allowing the maximum amount of light to reach the slide and the lens. Caution: do not use the diaphragm to control the light. It is to control resolution and contrast. Use the dimmer instead, or the light adjustment. Then, rotate the nosepiece so that the objective lens with the lowest level of magnification is directly above the sample. Reminder, using lower magnifications first helps to select the part of the specimen of interest and then adjust further. Look through the binocular eyepiece and adjust the diaphragm until the amount of light is satisfactory. More light is better than less light, but the comfort of the viewer's eyes should also be taken into account. Then turn the coarse adjustment knob until the specimen comes into broad focus. Caution: you should not use the coarse focus with a high magnification objective for fear of the objective making contact with the slide. Then, turn the fine adjustment knob until the specimen comes into sharp focus. Caution: you should not take a long time to find focus, otherwise the high magnification objective could also hit the slide. If you are having a difficult time to find focus, then restart with the lower magnification objective. The viewer should then be able to rotate the nosepiece to higher setting and bring the sample into more and more detail with a minimal amount of refocusing. Then use the stage control, the condenser, or the brightness adjustment to see the specimen more clearly. After using the microscope, be sure to keep it properly and safely. So, first lower the stage using the coarse adjustment. The stage must meet its lowest level, then return the objective into the lowest magnification, which is the scanning objective. Then turn off the switch and unplug the power cord. Then return it to its rightful place. So these are the example of an image of an specimen using the scanning objective with a total magnification of 40 times, while using the low power objective with a total of 100 times magnification. These are an example of an image of a specimen using the high power objective with a total magnification of 1000 times. Talking about magnification, what is magnification? So magnification is the ability to make small objects seem larger, such as making a microscopic organism visible. So in order to get the total magnification, you need to multiply the eyepiece magnification times the objective magnification. For example, the eyepiece has a magnification of 10 times, while the scanning objectives has a magnification of 4 times. How much is the total magnification of the image? You're right. 40 times. Resolution. When we talk about resolution, it is the ability to distinguish two objects from each other. For example, the resolution of a microscope is 0.2 microns. So in that case, the specimen, or for example, you are viewing under a compound microscope, a smear of red blood cells. So in order to distinguish two different red blood cells from each other, they have to be 0.2 microns apart. And that's all for microscopy. And for our next, photosynthesis, the chemical reaction, and the three different photosynthetic pathways. So photosynthesis is the process by which green plants and certain other organisms transform light energy into chemical energy. During photosynthesis in green plants, light energy is captured and used to convert water, carbon dioxide, and minerals into oxygen and energy-rich organic compounds. In chemical terms, photosynthesis is a light-energized oxidation reduction process. Oxidation refers to the removal of electrons from a molecule, while reduction refers to the gain of electrons by a molecule. In plant photosynthesis, the energy of light is used to drive the oxidation of water, producing oxygen gas, hydrogen ions, and electrons. Most of the removed electrons and hydrogen ions ultimately are transferred to carbon dioxide, which is reduced to organic products. Other electrons and hydrogen ions are used to reduce nitrate and sulfate to amino and sulfohydride groups in amino acids, which are the building blocks of proteins. In most green cells, carbohydrates, especially starch, and the sugar sucrose are the major direct organic products of photosynthesis. The overall reaction in which carbohydrates, represented by the general formula CH2O, are formed during the plant photosynthesis can be indicated by the following equation. So this equation is merely a summary statement for the process of photosynthesis. Actually involves numerous reactions catalyzed by enzymes. These reactions occur in two stages, the light stage consisting of photochemical reaction and the dark stage comprising chemical reaction controlled by enzymes. During the first stage, the energy of light is absorbed and used to drive a series of electron transfer, resulting in the synthesis of ATP and the electron donor reduced nicotine adenine dinucleotide phosphate or NADPH. During the dark stage, the ATP and NADPH formed in the light capturing reaction are used to reduce carbon dioxide to organic carbon compounds. This assimilation of organic carbon into organic compounds is called carbon fixation. As has been stated, carbohydrates are the most important direct organic product of photosynthesis in the majority of green plants. The formation of simple carbohydrates, glucose, is indicated by a chemical equation. So, little-free glucose is produced in plants instead of glucose units are linked to form starch or are joined with fructose, another sugar, to form sucrose. Not only carbohydrates, as was once thought, but also amino acids, proteins, lipids, pigments, and other organic components of green tissues are synthesized during photosynthesis. So for the final part of this topic, we will talk about photosynthetic pathway. So there are three types of photosynthetic pathway done. The C3 plants. So around 85% of plants in the world are C3. So C3 refers to the type of carbon molecule it produced during the process. C3 photosynthesis occurs in the mesophyll cells. During photosynthesis, the CO2 is taken up and transported directly into the Calvin cycle, where it is fixed into a 3-carbon molecule. Thus, where the name C3 comes from. If the stomata are open, CO2 moves in while oxygen moves out. But under a hot or dry condition where the stomata are closed, oxygen accumulates, which inhibits the amount of C3 that can be made. So, some plants evolve in adaptation that allows them to photosynthesize in a hot and dry condition. These plants carry out C4 photosynthesis instead of C3. So, during C4 photosynthesis, CO2 moves in the mesophyll cells and is immediately fixed into a 4-carbon molecule, thus the name C4 comes from. But the anatomy of the C4 plants is different from the C3 plants. In C4 plants, chloroplasts are found in the mesophyll cells and also in the bundle of sheath cells which surrounds the leaf vein. So CO2 moves from the mesophyll cells into the bundle of sheath cells where the Calvin cycle occurs. So this shields the plant from the oxygen buildup that would otherwise occur when the stomata are closed during dry condition. Addressing the problem of oxygen buildup is to change the timing of photosynthesis. This is called CAM photosynthesis, and in many ways it is similar to C4 photosynthesis except that the timing of process changes. So, during CAM photosynthesis, CO2 is taken during the night when the stomata are open and is fixed into a 4-carbon molecule and then stored until daylight, in which it enters the Calvin cycle. So, this adaptation allows photosynthesis occurs in drier climates. So, to summarize them, C3 is when CO2 is taken up and transported directly into the Calvin cycle, which is common in most plants, while C4 alters the location of photosynthesis. Example is the cord. CAM plants changes the timing of photosynthesis, which is done by the cactus. For our next topic, we will tackle genetics. So, we will talk about phenotype, genotype, law of independent assortment, law of dominance, and the law of segregation. Genetics is the study of heredity and the variation of inherited characteristics. So, how does these genes work? So, genes are instructions for building the parts of your body and doing the work that keeps you alive, from carrying oxygen to digesting food and everything else. So the term phenotype refers to the observable physical properties of an organism. This includes the organism's appearance, development, and behavior. While in a broad sense, the term genotype refers to the genetic makeup of an organism. In other words, it describes an organism's complete set of genes. So phenotype means the physical appearance, while genotypes refers to the alleles that makes up that certain appearance. So what does allele means? So it is a variant form of a gene. Some alleles are dominant while others are recessive. So, alleles can also refer to minor DNA sequence variations between alleles that do not necessarily influence the gene's phenotype. So, these are an example of homologous pair of chromosomes, and the colored parts are the locus for flower color gene. So, the upper one is the allele for a purple flowers, while the lower part is the allele for a white flower. So next is the principle of Mendelian inheritance. So first is the law of segregation, which refers to the two alleles for each gene are placed in different gametes. Law of independent assortment, the inheritance of one gene doesn't affect the inheritance of any other gene. Law of dominance, when two different alleles are present, only one is dominant and will be expressed. So, Gregor Mendel, 1822 to 1884. So, he is an Austrian monk who formulated fundamental laws of heredity in early 1860s. He studied science and mathematics at the University of Vienna, formulated the particulate theory of inheritance. So, inheritance involves reshuffling of genes from generation to generation. So, law of dominance. So, the law of dominance says that there are dominant and recessive traits. Dominant traits are defined as whichever phenotypes is expressed in an organism that is heterozygous for the trait. For example, the dominant phenotype are capital R represented by capital R and small letter r, while the recessive phenotype are represented by the small letter r. These are a good example of law of dominance. So, for the first cherry, the genotypes are homozygous dominant red. The second cherry represent with the genotype homozygous recessive, and the third cherry is represented by the heterozygous dominant. So as you can observe, since the capital A is dominant, which represent with the phenotype red, so when a recessive trait is mixed with the dominant trait, the dominant trait will be expressed in the phenotype. Next is the law of independent assortment. So the law of independent assortment says that genes for different traits segregate independently of each other. It means that separate traits are separately inherited. This is because during meiosis, the chromosomes line up randomly before the cell divides, allowing for gamete formation. So example, a true breeding round and yellow seeds crossbreed with a true breeding wrinkled and green seeds. So the first generation offspring will be all round and yellow seeds, but it doesn't mean that the wrinkled and green seeds are not present. It is just because they are recessive, so maybe they will be expressed in the next generation of offspring, but they are properly segregated. Next is the law of segregation. So the law of segregation says that everyone has two versions called alleles for each trait, one from each parent, and that this allele segregates randomly during meiosis. So these are example. So your father and your mother, example. So their genotypes will be segregated to all of you randomly. Next is the non-Mendelian inheritance. So the Inheritance of characteristics is not always as simple as it is for the characteristics that Mendel studied in pea plants. So each characteristic Mendel investigated was controlled by one gene that had two possible alleles, one of which was completely dominant to the other. This resulted in just two possible phenotypes for each characteristic. Each characteristic Mendel studied was also controlled by a gene on a different chromosome. So as a result, each characteristic was inherited independently of the other characteristics. Geneticists now know that inheritance is often more complex than this. A characteristic may be controlled by one gene with two alleles, but the two alleles may have a different relationship than the simple dominant-recessive relationship. These are the incomplete dominance and codominance. So incomplete dominance is when there is a blending of two alleles that resulted in a third phenotype that doesn't look like either of the parents, while in codominance, both alleles are expressed together in the offspring. Example: incomplete dominance. When a red tulip is crossbred with a white tulip, the result is a pink tulip. While for the codominance, when a red flower is crossbred with a white flower, the result is a variegated flower. Another example is for a cow. A white cow is crossbred with a brown cow. So the result will be a polka dots cow. This is an example of codominance. So these are examples of a incomplete dominance. So when a red flower is crossbred with a white flower, the offspring will be pink flower. So next I will teach you how to do crossbreeding using a Punnett square. So a monohybrid cross. The trait would be seed shape. So the alleles are capital R for round, small letter r for wrinkled. So the cross is round seed times round seeds. But the round seeds are a heterozygous dominant, both of them. So the results are shown below. So the genotype of the offspring are dominant, homozygous dominant round, heterozygous dominant round, and a homozygous recessive wrinkled. So the phenotype would be round and wrinkled. The genotypic ratio is 1:2:1. Why 1:2:1? It's because in the offspring, as you can see in the boxes, there are one homozygous dominant round, two heterozygous dominant round, and one homozygous recessive wrinkled. For the dihybrid cross, so dominant and recessive, capital T represent tall and capital B represent black color. These are the dominant traits. Small letter T represents short and small letter B represent white, and these are the recessive trait. So the crossbreed will be heterozygous dominant, tall and black, both of them. So how to do the dihybrid cross? See the line on how I do it. And observe how it is done. So these are the fork method. another method of crossbreeding. Then, last topic would be fertilization. The four major events of fertilization and the fertilization strategies. So, fertilization is the fusion of haploid gametes, a sperm and an egg, to form a diploid zygote. So, it accomplishes two things. It provides for the recombination of the paternal and maternal genes, restoring the diploid chromosomes, and it activates the egg to begin development. Fertilization marks the beginning of the development. So, in humans, it happens in the ampullary region. It is the widest part close to ovary of the uterine tube. Sperm cells may remain viable in the female reproductive tract for several days. So, in the ampullary region, it is where the egg meets the sperm before it goes to the uterus. So, the four major events in animal fertilization. First is the contact and recognition between sperm and egg. So, the egg releases a certain chemoattractant that attracts a certain sperm which fertilizes the egg. So, second is regulation of sperm entry into the egg. Third is the fusion of the genetic material of sperm and egg. Fourth is the activation of egg metabolism to start development. So, next is the fertilization strategies. So, external fertilization occurs when sperm and eggs are deposited in water. Example, the frog. The female frog deposited its eggs into the water, and also the male frog deposited the sperm into the water, and it is where the sperm fertilizes the eggs. Next is the internal fertilization. It occurs when egg is retained in the body of the female and the sperm are deposited there by the male. So most animals do the internal fertilization. Three strategies in internal fertilization. Oviparity. Eggs are fertilized internally and deposited outside mother's body to complete development. Examples of these are birds, reptiles. Viviparity. Young develop within mother and obtain nourishment directly from mother's blood. Example, most cartilaginous fish, some amphibians, a few reptiles, and almost all mammals. Ovoviviparity. Fertilized eggs are retained within mother to complete development, but all nourishment gained from yolk sac. Example are guppy or the Rhabistis reticulatus, some sharks, some lizards, garter snake or the Thamnophis sartalis, and some vipers. So, some animals will lay eggs and the fetus will develop outside of the female. They are called monotremes. While some mammals carry their young inside a pouch, they are called marsupials. Other mammals will continue to have the fetus develop inside the female until it has developed into a mature offspring via gestation. Next is the stage clip. So, this clip holds the slide in place. So, the gestation of some animals in months. So, the rabbit is 1 month, cat is 2 months, dog is 2 months, lion is 3.5 months, pig is 4 months, sheep is 5 months, human is 9 months, cow is 9.5 months, horses are 11 months, while the elephant is the longest, which is 22 months. And that's all for biology.